All right. Well, it is 12 o'clock on the dot right now. We can go ahead and get started if everyone is ready. So good afternoon, everyone, and welcome to our Department of Medicine Grand Rounds. My name is Ryan Ave. I'm one of the Internal Medicine Chief Residents, and today we have a special joint Grand Rounds, the Department of Psychiatry and Behavioral Sciences. So I have the pleasure of turning over the stage to Dr. Joseph Ceramelli, who will share more about our day today.
Thank you, Dr. Abe. Hello, my name is Joseph Ceramelli, and I oversee Grand Rounds for the Department of Psychiatry and Behavioral Sciences. It's great to hold a second joint medicine and psychiatry Grand Rounds. We did this once last year, which went well, and I believe such joint efforts can really benefit both departments. Importantly, today's Grand Rounds presentation is sponsored by the Garvey Institute for Brain Health Solutions. And this is a University of Washington-wide institute committed to developing, testing, and rapidly deploying innovative approaches to prevent and treat a range of brain health and mental health problems. Now, there are people actually from both medicine and from psychiatry working in the institute, and such as on advisory groups and on innovative grant projects. So thank you to the Garvey Institute for sponsoring today's Joint Grand Rounds presentation. And now I'll turn it over to Dr. Unitzer.
Okay, well, good afternoon, everybody. Thanks for getting us started. My name is Jürgen Unitzer. I chair the Department of Psychiatry and Behavioral Sciences at UW Medicine. And today I have the honor of introducing our speaker for this Joint Rounds presentation in medicine and psychiatry. And as Joe said, this presentation is sponsored by the Garvey Institute for Brain Health Solutions here at UW Medicine. And I want to give a big thanks to Lynn Garvey and her daughter, Denise, who are joining us in the audience today. So our speaker today is Dr. Emron Meyer. I'm going to tell you a little bit about him. So Emron was born in a small town in Bavaria, where his family ran a confectionery business since I think in the 1800s. But instead of following the family business in nutrition, he went to medical school at the Ludwigs-Maximilian-Universität in Munich. And that's incidentally the same university where Dr. Jung and I also started our academic trajectories. After medical school in Munich, he trained in medicine at the Vancouver General Hospital and then ultimately found himself at UCLA, where he has had a really outstanding career as a gastroenterologist. But there's a lot more to Dr. Meyer. He has a passion for mountaineering and for filmmaking, and he's had some really interesting adventures in film and mountaineering that he probably won't tell us about today, but we got some great mountains here, so maybe at some point we can entice him to come and explore a few of those with us. I also love mountaineering. In his work, he's explored ancient and really diverse healing practices around the world, and he works to integrate the wisdom from these traditions into his research and his clinical work. Apologies. Today, Dr. Meyer is a distinguished research professor in the departments of medicine, physiology, and psychiatry at UCLA. He directs the Center for the Neurobiology of Stress and Resilience, and he's the founding director of the UCLA Brain Gut Microbiome Center. He's published hundreds of scientific articles and also two really great popular books. I see them right behind him. On the connections between the gut, the mind, and our immune systems. I think he's one of a handful of individuals who's really spent much, if not most of his career working at this interface. And that's an area that I think is rapidly becoming one of the most exciting topics in medicine and healthcare today. So why have we asked Dr. Meyer to present to this joint audience in medicine and psychiatry? Well, in addition to being an accomplished GI doc, Dr. Meyer is a highly accomplished neuroscientist. In his research, he asks questions such as how the chronic GI inflammation relates to changes in our brain. How does the food we eat and how do our microbiomes affect what happens in our brains, how we think, how we feel, how we act, and how we regulate our emotions? Those are questions that when I was in medical school, I don't know, sure anybody was asking those questions, but Emron has actually been asking those questions for a couple decades. And so it seems that he has in fact followed his family tradition and his childhood roots in nutrition, just in a different way. I think he's truly one of a kind, a true pioneer. I think the perfect speaker for a joint grand rounds in medicine and psychiatry. So let me conclude on a little bit more of a personal note. I think for Barbara Jung and I, it's a great pleasure to welcome somebody who shares many of our childhood roots in Bavaria and somebody who bridges our two worlds so nicely. So please join me in extending a very warm welcome to our speaker today, Dr. Meyer. And now I'm going to ask Barbara to follow with a few words.
Thank you, Jürgen. Well, very tough act to follow. I would have said it more directly. Jürgen does the brain, I do the gut, and Emron connects the two. So I couldn't be more pleased. Just to build a little bit on what Dr. Unitzer was saying, with the Garvey Institute for Brain Health, we have an amazing opportunity. At the same time, we're working on transforming our microbiome center. And again, nobody better than to kick us off with a joint grand rounds. I'm going to leave it at that. We've had a lot of really interesting talks around what to do next. And I can't wait to hear Emron showing us what he's working on right now and sharing it with a broader audience. So welcome, Emron, and thank you for being here.
Yeah, thank you, Barbara, and thank you, Jürgen, for the invitation. This is really, it's a pretty unique coincidence that all three of us come from a very similar, closely adjacent region of southern Germany. So I have to say in my career, this has never happened before. So I really appreciate that. And thanks for the kind words. It's been interesting. I mean, for some reason, I've had this interest of connecting things from the time of my medical school. And I still remember when I was looking for a thesis advisor at the time, I went from one professor to the other and told him I would like to study the interactions between the brain and the organs. And so the most common answer I got is Dr. Meyer, we all know that psychology plays a role in medicine, but it can't really be studied. So I was lucky. I did find somebody through some interesting interviews from the Max Planck Institute to finally Department of Physiology and started my career in studying the connection between the brain and the heart. So the influence of stress and emotions on regional cardiac blood flow. And that sort of launched me on that path, and found I spent 4 years working on this. Unfortunately, this is not recognized as a real PhD in the US, but it certainly was the equivalent in the basic science department. And anyway, so I have applied this idea of connecting things that normally in medicine are not connected from the heart onto the GI tract. And I'm glad I did that, because what turns out to be the case that the brain-gut axis is clearly, now one of the hottest topics, both in research, but also in clinical treatments. So now let me just pull up the 1 second. Okay. Yes, I said, so these are the entities, commercial entities that I'm on the scientific advisory board of. These are the topics that I want to address, some very briefly, some a little bit more detail. So a few overview concepts about the brain-gut microbiome system, then focus on the neuroactive signals within that system, the immune interactions. So a few words about the role of the gut microbiome in chronic brain disorders. Then a topic that I find very intriguing because it fits both the brain and the immune system very well together. So this influence of chronic environmental and dietary stress on the immune system. Say a few words about diet, and then a take-home message. Now, each of these topics could be a separate talk, and I hope the short versions I'm presenting to you will give you sort of a general idea of this field of medicine and psychiatry. One way, so initially this, and personally I also refer to this as the brain-gut axis, the brain-gut microbiome axis. I have replaced this with the brain-gut microbiome system. The reason is because you really need a systems biology viewpoint and perspective to understand how it works. So there are multiple bidirectional loops between on one side between the brain and the gut and its microbiome, but there's also intermittent loops with the immune system, and there's loops with the environment in multiple ways, both in terms of mediating psychosocial influences, but also environmental influences in terms of soil, water, air, pathogens, and antibiotics. And any system you study or you understand, if you can perturb it, perturbation is a stressor. So psychosocial stress, obviously directed at the brain, and then reverberating throughout the system, and dietary stress focused on the gut and the microbiome, and again, reverberating it. Some of these effects, as you'll see in my presentation, seem to be going in a very similar direction. So if you're exposed to both kind of stressors, you seem to be getting the amplified so-called allostatic load on the system with its consequences of chronic disease. So everybody knows this in the meantime. So 40 trillion microorganisms, archaea, fungi, viral organisms, and bacteria in our gut. About the same number, plus minus, of our human cells. What's more interesting and really relevant is that the comparison at the genome level, 20,000 human genes, which are actually, very similar between humans, even between humans and primates, non-human primates, even mice, compared to the 2 to 20 million microbial genes, the function of most of which we don't really know yet. So we neglect about 99% of the system in our pathophysiology, in our textbooks, and in our treatment approaches. I think this is sort of the biggest reason for being excited about this field, because there will be many unexpected findings coming out of it. So the gut microbiome, and this is, the center of this talk, is not restricted to the lumen of the gut. First of all, there's other microbiomes on the skin and on every epithelial surface. One is that it has close interactions with the gut, and we'll be talking about this, and with the immune system. Another one, it plays a significant role in shaping our immune responses for later in life in terms of food allergies and autoimmune diseases. It has a major influence on the liver. Metabolism or metabolic disease-related liver diseases such as NAFTAL have been shown to be related or associated with dysbiosis or alterations in the microbiome. Cardiovascular disease, Tmao, has been identified as a major risk factor for cardiovascular disease, and it's produced by certain microbes from certain food components, then further metabolized in the liver. And depending on your gut microbiome, you will produce from carnitine, for example, containing food items, more or less of this substance. And then, so the interest that has the area that's certain, my main interest is really with the brain. This started out with some really, really spectacular studies in mouse models, shown showing that there's a relationship of the gut microbial composition and its metabolites with brain structure, with brain function, and with emotion regulation. And this has been extended to other disease models from Alzheimer's to autism spectrum. One thing I should emphasize through all of this is many of these links have been shown in terms of associations. The biggest gap in our knowledge in this field is really understanding causality in which of these diseases does the gut microbiome and alterations such as so-called dysbiosis play a causative role in these diseases and is a target for novel treatments that we have not thought about before. So there's a few schematics now. One is going to go over obviously the microbes need to have molecules with which they signal to the body and to the brain. And you can break this down into 3 categories of these signals. Start out with the ones that probably have received most of attention that are diet derived. For example, the dietary fiber and polyphenols, these very large molecules that cannot be absorbed in our gut intact, but travel down into the distal small intestine and colon to be broken down into smaller components. The fiber molecules into short-chain fatty acids. That category of signaling molecules has received a lot of attention because they have anti-inflammatory effects and act pretty much have receptors on every cell type from the gut, enteric nervous system, vagus nerve, and the brain. You could almost call this it's the pharmacy that our microbes help us to produce to regulate or suppress or downregulate inflammatory processes throughout the body. Then there's the amino acids also, and I'll show you some examples of each of these. Breakdown products into the indoles are particularly interesting categories. We know a lot about this. We know also, so tryptophan is one of those molecules that have best been studied in terms of breakdown products, serotonin, indoles, and kynurenines. Then we have another interesting group of molecules that get into the gut and are then processed by the microbes into signaling molecules, primary bile acids being secreted with the bile into the gut, and then processed, metabolized by different communities of microbes. Everybody has different composition of these communities, because there's a large number of different bile acids. Depending on the profile of these secondary bile acids, they have either a net positive effect on the nervous system or have a negative effect. So abnormal ratios of secondary bile acids to primary bile acids have been reported in cognitive decline and in Alzheimer's disease as one factor. Another very interesting but poorly studied field is steroid hormones. So female sex hormones, estrogens, are also metabolized in the liver or modified in the liver, excreted in the bile. And again, depending on somebody's specific ecological system of microbes, processed into several metabolites, many of which are absorbable. So then these estrogen metabolites are reabsorbed, enter the systemic circulation, and can play a significant role, for example, in how a woman does after menopause. This is also important for the processing of external, so-called phytoestrogens. For example, in tofu, not everybody converts those into estrogens, but people that have these communities, they will do that. And then there is de novo synthesis and generation of chemical signals from, for example, from the membrane of microbes, the so-called MAMPs, LPS falls in this category, and toxins. So a lot of toxins that we ingest by themselves may not be toxic to us, but once they're processed by the microbes, develop an ability to harm mammalian cells. So this has been shown, for example, for glyphosate, which by itself does not seem to have negative effects on the human host, but the metabolites do. So we have these three categories, and then we have these three modes of transmission of these signals. So one is neurocrine, acting locally on afferent nerve terminals. I'll show you this in a minute in a picture. So many of these microbial generate metabolites have their main receptors, like short-chain fatty acid receptors, secondary bile acids, on afferent vagal nerve terminals, and are able to signal to the brain. So this is the Vagal Efference story. There's also serotonin, which is synthesized and stored primarily 95% in the gut in specialized cells, and which can act on the enteric nervous system directly. So then there's the endocrine fashion. So these are metabolites that are released into the systemic circulation. They are absorbed and get into the systemic circulation, interacting distally. So satiety signals, GPL, a series of satiety signals that are located in enteroendocrine cells in the distal small intestine. They have receptors on the surface, these endocrine cells or enterochromaffin cells, that have an influence both on the synthesis but also on the release of these signals. There's also one of the metabolites from tryptophan metabolism, kynurenine, which can pass both the gut vascular barrier and the brain, the blood-brain barrier, and get into the brain. And there's also the indoles. So this can act through the systemic circulation. And then there's systemic immune mediators that can act locally. Also, many of these receptors, such as the toll-like receptors, being present on cells in the gut, also on vagal nerve terminals, but they can also get into the systemic circulation, either in terms of intact microbes that can pass this gut vascular barrier, but also in terms of cytokines, MAMPs, and LPS. To say a couple of words about gut microbial signals, I mentioned tryptophan, probably one of the best studied and fascinating candidates of that. So this is an enterochromaffin cell in the gut. So they are sandwiched in between the gut epithelial cells. It's become known in the last few years that they have these extensions, these neuropods. So these are not just cells sitting there in isolation, but they have these extensions which form synaptic connections with, for example, vagal efference and probably spinal efference. And they also have synaptic connections with, for example, sympathetic and vagal efference. So they're almost like an extension, these cells, of the brain with a very close regulation. But most importantly is that the rate-limiting enzyme for serotonin, TPH1, is regulated, at least in the mouse, to about 40% by signals from certain gut microbes, the Clostridialis, through short-chain fatty acids, secondary bile acids, with their specific receptors on the luminal side. And then interestingly, these cells not only store most of the serotonin in our body, they signal to the brain in a bidirectional way, but they can also release serotonin back into the lumen and influence the behavior of the microbes. It's also been shown by Lane Chao at UCLA. She's a member of our Brain Gut Microbiome Center, that some microbes, Clostridialis, again, have the serotonin transporter molecule in their membrane, so they can actually take up that serotonin, and it changes their behavior. Exactly what function that has, we don't know, but when you think about the effects of antidepressants, they certainly affect those microbes with their transporters as well. Then there's another group. So most of the tryptophan is actually metabolized to kynurenine. This is also modulated by microbes. It's not synthesized, but it's modulated. So lactobacilli from a mouse model of chronic variable stress produce hydrogen peroxide, which inhibits the rate-limiting enzyme for kynurenine synthesis from tryptophan. So if you have sufficient number of lactobacilli, you will inhibit this transformation. If under chronic stress, there's a decrease in lactobacilli, there's a disinhibition, and you'll now generate more kynurenine. So kynurenine in general has negative effects on various in vitro and in vivo neural systems. It enhances like NMDA receptor stimulated excitotoxicity. It plays a role in neuroinflammation and also in neurodegeneration. So this just gives an example that the number and composition of these two groups of microbes, the Clostridialis and the lactobacilli, can have very different effects on the outcome from what happens to ingested tryptophan. And then there's a third group of molecules, the indoles, and they are not just modulated by the microbes, but their metabolism is totally dependent on microbes, because only microbes have the enzyme that leads this transformation from tryptophan into indole. Indoles, so it's a large family of molecules. One of their metabolites that is generated in the liver after all these subforms, all these subtypes of indole metabolites are being generated. Indoxyl sulfate has been implicated in various processes, not just in the nervous system, but also, for example, in kidney failure. So it's clearly an indicator of degenerative inflammatory processes. So this gives you some sort of an idea of the complexity. It's just one amino acid, and we just talked about one of these metabolites. Each of these metabolites occurs in multiple subforms, and this is regulated by the dietary intake of tryptophan. For example, this stimulation depends on how much tryptophan that you ingest in your diet. But just giving a serotonin supplement would obviously be a very, very small part of that effect, and therefore it's probably not become a commonly used treatment for depression or other psychiatric diseases. A few words about gut microbiome immune interactions. So if you look at the intestine, there's several barriers. So you can imagine 40 trillion microbes with the gram negatives having molecules like LPS in their membrane, with LPS stimulating toll-like receptors. If the fact that the system is only microns away from our immune system is a pretty amazing evolutionary accomplishment. And the way it's accomplished is that there's several barriers. One is it's not really illustrated here, is a mucus layer that separates the microbes in the gut from the epithelial layer. Then you have this epithelial. So this is pretty tight in a healthy gut and impermeable. Then you have this epithelial layer with these tight junctions between it that prevent, under normal conditions, non-inflammatory conditions, any translocation. And then you have the gut vascular barrier, which essentially is very similar to the blood-brain barrier that normally is not permeable to the microbes or to many microbial substances. So any compromise in the system, either in the mucus layer, in these tight junctions, or in the gut vascular barrier, will lead to an exposure of immune cells to pro-inflammatory substances, and the activated immune cells will produce cytokines, and from this innate immune system will activate the adaptive immune system, and this will go, once it passes this barrier here, go into the systemic circulation. It's also been shown that bacteria can, both our normal microbes in animal models are important in assuring a tight blood-brain barrier, but with inflammatory influences, this is loosened, just like the gut epithelial and the vascular permeability are loosened with inflammatory mediators and can lead to neuroinflammation, and if it's chronic, to neurodegeneration. So there's two connections. We talked about the neurocrine communication. So one is the vagal pathway, and one are the dorsal root ganglia pathway. And I won't get into this in detail, but activation of immune cells can both affect the vagal efference, but also extensive literature on peripheral sensitization of spinal efference, which then lead to enhanced transmission to the dorsal horn, and secondary sensitization, central sensitization of pain pathways. And like everything in that system is always bidirectional. So vagal influences can also have an effect on macrophages, on dendritic cells, usually in form of an anti-inflammatory effect. So inflammation can engage these systems generally in a negative way with symptoms such as activation of the HPA axis, sickness-related behavior, kind of an animal model of depression, increased pain perception, but activation of certain vagal pathways can downregulate inflammation in the periphery. So we talk about vagal efference, and this is another area that Elaine Shao and her group is working on. I think it's really important to remember there's not just one afferent vagal nerve or nerve endings. The vagus has very site-specific. So this is illustrated here. You know, the innervation density is the highest in the small intestine, is also present in the stomach. You know, we know this from the whole gastric ulcer story, but it decreases in the large intestine, pretty much absent in the distal large intestine. Also, sub areas in the gut are innervated by different vagal efferents. So blood vessels, endocrine cells shown here, and the two muscular layers and the mucosal layer. So very complex system, which we're just beginning to understand. So this shows you, look at the nucleus tractus solitarius and nodus ganglion. This is where this influence from the gut, the vagal influence, converges. But you can see this illustrated here. Each of these subtypes of fibers has a different phenotype in terms of the receptors that are present on them. Each one is different. I don't know how many different. I think there's 12 or 20 subtypes of vagal afferent receptors. Each has different receptors on them, responding to different molecules that come mostly from the microbes. So a very complex system. What we've done in the past, for example, with peptic ulcer treatment, we just cut not just the sensory part of the vagus, but also with a truncal vagotomy, the entire vagus. It's amazing that was really consistent with survival and health. But many people developed really strange symptoms. I still remember that time from early in my career. Unfortunately, we didn't have the knowledge to understand why they developed very strange psychiatric symptoms. One syndrome, the Albatross Syndrome, that people never were the same as they were before the truncal vagotomy. We will never have a chance to restudy this because obviously these surgeries are no longer performed. So once it gets to the brain, there's also a lot of things that these microbial signaling molecules that we first went through, the different categories, what effects they have. So if you look at these membrane components, MAMPs, or also the LPS, neurotoxin neurotransmitters lead to neurodegeneration and neuroinflammation in neurons, but also in glial cells, oligodendrocytes, astrocytes, microglia, and also in endothelial cells. The thing that I want to highlight here is, first of all, there's in this, the detrimental effects include almost in all of them these membrane components of gram-negative microbes, and they also include neurotoxins. The mechanism, all these areas include all these pathways include inflammation and neurodegeneration. This leads to a weakened blood-brain barrier. So there's some general concept here that microbial signals, primarily these immune related signals that stimulate immune signal in the gut, have an effect on the immune system within the brain, the glial cells, and also almost certainly play a role if these changes are chronic in neurodegeneration and diseases that are characterized by neurodegeneration. Now, the beneficial ones, I just want to emphasize the similarity here. You see that short chain fatty acids are present in all of them. So I was telling you earlier, this is almost like the pharmacy of the microbes, the aspirin that the microbes produce, so the NSAIDs, that this reduction of inflammation is a major function of these metabolites that are generated from the breakdown of complex plant-based carbohydrates that have this inflammatory effect. So you get some idea here why, and I will come back to this at the end, why a plant-based diet has all these reported beneficial associations with brain health. It's not a coincidence that both polyphenols coming from plants and short-chain fatty acids are some of the major mediators. So there's many brain disorders, many chronic brain disorders where an alteration of the brain-gut microbiome system has been implicated. And as I said before, in most of them, it's still demonstration of an association. There's some causality from animal models, but in humans, most of it is association. The closest to causality is going from a mouse model, or going from transplantation of altered gut microbiome in patient population into germ-free humanized mice, and then looking if this mouse model then develops human-like disease features like Parkinson's or depression-like behavior. And without having time to get into the details of it, so autism spectrum, there's a lot of evidence, and since this is a developmental disease, it doesn't really start, 1 or 2 years after the infant is born, but it starts in utero. Then activation of the gut-based immune system from an altered gut microbiome in the pregnant mother, leads to immune signals that affect the development of the fetal brain. So all the animal models are based on that mechanism, the maternal immune activation model. Multiple sclerosis, again, this has been associated with an alter-programming, like other autoimmune diseases, an alter-programming of the immune system by the gut microbiome early on in life, and an imbalance between in the immune system between pro-inflammatory and anti-inflammatory cytokines. Stroke, very interesting story that the infarct size is related to the state of dysbiosis in animal models, and that the dysbiosis has an influence on inflammation in the brain after an experimental infarct. This has not been demonstrated in humans, but the animal findings are pretty intriguing. Parkinson's disease, again, starting in the gut in a large number, probably in the majority of patients, in the enteric nervous system, were the same alterations. These Lewy bodies have been demonstrated in the enteric nervous system first, and then seem to be traveling up the vagus nerve into the central nervous system and leading to microglial activation. So inflammatory bowel disease, I won't get into this, but clearly evidence as well. And then obesity and metabolic disease in general has been associated with these systemic immune activation, low-grade immune activation, and changes in the sensitivity, for example, of satiety receptors in the hypothalamus. So I'm probably gonna end after this section. This to me is sort of really intriguing. If you compare the healthy gut with what's been referred to now in the lay press, but also in some scientific talks as the leaky gut, so an increased with a decreased gut barrier, increased permeability, and normally people don't differentiate the mucus layer part and these three different mechanisms. With a healthy diet, we have a high diversity of gut microbes in the gut. These certain of these microbes play a crucial role in the generation of an effective mucus layer that separates this healthy microbiome from the surface of the gut, particularly from dendritic cells that have their extensions sticking into this mucus layer. And the minute they come in contact with a microbial surface, it triggers these toll-like receptors on these cells and then triggers the whole cascade of immune activation. So healthy diet, increased high diversity, strong mucus thickness, tight junctions between the epithelial cells and intact gut barrier. With chronic dietary stress, this means in this context really the standard American diet, high in fat, high in carbohydrates, low in plant-based fibers and polyphenols, high in additives, leads to a decrease in the diversity, decrease in the mucus layer thickness. Now these microbes, some of these microbes come in contact with the dendritic cells, stimulate cytokine release. Cytokines loosen these epithelial tight junctions. And then there's the translocation of microbes or microcomponents into the systemic circulation, leading to something that's been called metabolic endotoxemia. So an endotoxemia without infectious organism. Now, this probably happens in a lot of people. I often, when I give a talk, I look at the audience and say probably about 40% of the audience will have some form of metabolic endotoxemia. Certainly, if you're overweight or obese, you fall in that category. But not everybody gets the disease. So many people, well, if they're not tested, they will never know life. But if this combines with genetic risk factors for any of these disorders, your risk will greatly be increased to develop them. And so that's sort of, I think, a big target now of research that will make a big difference to test that early, both the genetic risk factors, but also the presence of this of this unhealthy situation, immune activation. So this state of dysbiosis, this decreased diversity and disappearance of certain microbial strains, has really been going on in the last 75 years with industrialization, westernization of developing countries. Some of the factors are shown here, diet being a big one, chronic stress being another one, programming of the gut-based immune system and the microbiome by the changes in colonization, delivery in the hospital, antibiotics during delivery, and altered exposure to antibiotics early on in life to a large number of infants and particularly, preemies. So the gut microbiota are very quick to adapt to these altered environmental factors. I told you this, they have 20 million genes. So their ability to adapt our organism rapidly to this new environment faces something else, which is our 20,000 genes, something that's very stable. It takes, decades for at least for human genes to change. So our own gut and immune system is much less adaptable. So you get this mismatch between this significantly altered microbiome and a conservatively maintained gut. The gut perceives this as a perturbation, as a stressor, and reacts with engagement of the immune system. And that in turn then leads to this metabolic endotoxemia. Share a couple more different presentations to emphasize this. So we have again the brain, the microbiota, and the gut with these bidirectional communication pathways. We have two types of chronic stress. One is targeted at the brain. You could also say in the last 75 years that chronic stress has increased dramatically in the last 2 years with the pandemic. And then we have the dietary stress, or the diet that has led to rapid changes in the gut microbiota, but is perceived by the gut and the gut's immune system as a major stressor. And so chronic stress that engages the physiological, evolutionary developed stress systems in our body has been referred to as allostatic load. So evolution did a phenomenal job in creating our normal stress response, with HPA axis activation and sympathetic nervous system activation, but it did not develop optimally or adaptively to a situation of chronic stress. And the same can be said about the diet. Our gut has not developed to see the kind of things on a chronic basis. So, our gut is very good in adapting rapidly to any kind of change in the diet. So people have survived starvation, survive major infections. These are all major stressors on the system, but it's not well developed to deal with chronic stress. So the allostatic load on the system, both from psychological and dietary stress, is something that, in my opinion, is really at the core of this epidemic of non-infectious diseases that we have seen in statistics. We don't have the causality normally in general for these connections, but we have a lot of associations that paint a very good picture. And we also know that the main pathways by which this gut health and the gut-based immune system has been affecting the function and integrity of all our organs. So I think I'm going to go to the end. So this is something that in my last book I sort of came up with. I have to say this is connecting another component, sort of following my obsession with connecting seemingly unrelated areas. So clearly the microbiome plays a major role in human health, but which I couldn't show you, it plays a major role in plant and soil health. There's a whole literature exploding in this area now that the health of the plant and the nutrient content of plants is strongly dependent on the health of the soil microbiome. And if you eat plants that come from an unhealthy soil with compromised soil microbiome, it will not have the beneficial effects, for example, polyphenol mediated, as something that has grown in a regenerative organic soil. So there's some, far-looking individuals, visionary individuals that are pushing this agenda, both in industry and some companies. I shouldn't make any commercial here, but Patagonia and Yvon Chouinard, the founder, is sort of, even though he's in his early 80s now, is an inspiring figure who keeps promoting this concept in his business operation. So to finish, this communication between the gut microbiome and the gut-based immune system is an ancient evolutionary conserved system that modulates the function of every organ, including brain structure and function. It connects the mind, the gut, connectome, food, and the environment in a way that can best or only be understood in a systems biological way and modeled with network science. Certain microbial taxa hold keystone positions in these networks, just like in any other ecosystem. And key components of plant-based diets act through modulating keystone species of the gut microbiome to reduce systemic immune activation and the prevalence of chronic infectious diseases. So this is our group and our funding. And anyway, if you want to learn more about this, subscribe to our newsletter, go to our website, or you may want to take a look at one of these two books, which lays it out in much greater detail. Thanks very much for your attention.
Thank you so much for just an excellent and interesting presentation. I wanted to open up the floor now for a Q&A for our speaker. Please post any questions in the chat. You have a couple that have come through already. Let's see. One question is that you mentioned that medical practice can also lead to dysbiosis and included vaccinations in that. Could you clarify that point a little bit more?
Yeah, so vaccination, obviously, that's a hot potato. There's certainly no good evidence at the moment that vaccination plays a significant role in this. I discussed this with a key opinion leader in this area, Rob Knight at UCSD. So if in the future we'll find connections, it's always possible, but at the moment it's not. It's really, I think on the top are the antibiotics, both in early life, during pregnancy, during delivery, even from animal models. A one-time dosing in the pregnant mother can have a negative effect. But there's also other medications. I told you that, for example, antidepressants have an effect on microbial, behavior, and there's probably most medications are in some ways, affect microbial function and composition. So I would say this is also a new area that will, provide a lot of information that we didn't even think about before.
Dr. Meyer, a question about assessment, and thank you for this presentation and outlining these connections. Regarding the dietary and psychological stress, it seems like psychological stress is just easier to assess for, to ask people directly about it. How does one clinically assess for dietary stress? Is it simply asking, what do you eat, or is there another way?
Yeah, so traditionally, it's been through questionnaires. There's food questionnaires, there's interviews. In general, they're not reliable, even though they're now used in most microbiome studies. There have been studies comparing questionnaire-based assessment with actual intake, and there was zero correlation, which is really interesting for interpreting studies. So there's a new development. It's called Foodomics, driven by Peter Dorestein at UCSD, where they tried to identify metabolites of different food components, both in the stool but also in the plasma. So here's some pretty interesting results already. They believe that in a couple of years they will be able to completely replace any questionnaire-based assessment that you can essentially with measuring a very large number of metabolites and using artificial intelligence to determine what this person is eating. So the answer is it can be assessed. Multiple, the FFQ, food frequency questionnaire, we've been using in the interview, but when you hear these reports that there's zero correlation between what individuals tell you, and it's not done on a they don't want to lie. It's just very difficult to remember exactly. You probably want to talk less about the unhealthy things, and but that will change. I mean, that's going to be a major breakthrough. I think this foodomics approach.
We only have about 2 minutes left, so I'll just pose one kind of double question, try to get some of the questions that are coming through in the chat. Are there any training resources for nutrition for clinicians that you'd recommend? And are there any strategies that can help rebuild gut microbiome after antibiotics or other sort of dysbiosis triggers?
Yeah, so the microbiome is pretty resilient, like most ecosystems. If you start out with a compromised one that get all the hits during development, from the pregnant mother to the infant to being a preemie, then obviously your microbial ecosystem is less resilient, and when you take an antibiotic as an adult, it's much more likely to cause problems, and it doesn't bounce back. But there are studies where people have been given 3 consecutive doses or courses of antibiotics, and it came back to the normal level. So it depends, a big inter-individual difference. To rebuild it, I think at the moment the only thing that we have, and it's kind of ironic, there's all this amazing science that is comparable to going to Mars, in its complexity. But we don't really have anything yet that we can say, we intervene better than with a healthy diet. So, I mean, the healthy diet remains at the moment the only thing that, and the avoidance of, the healthy diet means avoidance of emulsifiers and chemicals in the food and ultra-processed food. Learning this, so there's several books out now. Fellow gastroenterologist, Will Bulsevich, so he pushes a vegan diet and got a big name recognition on his fiber-fueled book, fiber-based. I love the polyphenols because they're much less well understood and less studied. So yeah, there's several books out there. There's also a lot of confusing stuff out there, that in my book, I had a whole chapter about diet and recipes in the second book. But there's many other resources. I don't think there's specific courses for physicians, which is kind of surprising. Considering how important this is becoming, this should be mandatory almost. Certainly for a gastroenterologist, I think to have a basic education in this field.
Well, I think on that note, Dr. Meyer, we will close out mandatory cooking classes for physicians to help their patients. And I'm only half joking. Thank you so much for coming to us and bringing your transformative ideas. This is not the last we're going to hear about this. You gave us a lot of ideas on what to read and what to think about, and particularly when we prepare our foods and think of the foods of our patients and our brains. So thank you so much. And thank you to everyone who joined us today.
Thanks again. It was a pleasure. Hope next time we'll see you face to face in person.
That sounds wonderful. Goodbye, everyone.
Okay, bye-bye.